Vertebrate Zoology, coming at you with the last lecture for Exam 3. This is on Chapter 15. Chapter 16, which is on the hexapods, will be on Exam 4. We're a little behind schedule because I really like spiders and I wanted to include a lot of pictures of them, so this one took me a little longer than normal. I've got my scorpion collar dress on today because we're talking about the Chelicerates, one of my favorite groups. I mean, honestly, they're all my favorite groups, you know that, but let's get into it. Okay, obviously I had to include this picture by Ernst Haeckel showing a little bit of the diversity that are in the subphylum Chelicerata. And again, make sure you download off of Schoology your learning goals and fill in that study guide while you're watching the lecture. So let's talk about chelicerates. There are about 75,000 described species. That includes things like our cutie pie pet horseshoe crab, Shelly, that we have in our marine invert tank. I took this cute little video of her eating some shrimps. It also includes sea spiders, scorpions, and things related. This is not a scorpion. We'll talk about it. And then it also includes spiders, mites, and ticks. This is one of my favorite species of mites. Well, that we'll also talk about. The two defining features of this group that almost all of them share is a lack of antennae and then a set of chelicerae. Chelicerae are the only head appendage that's anterior to the mouth parts, and they're usually pincer-like claws in most of these groups. They have two distinct regions. There's the prosoma, and then there's the opistoma. Don't know if I'm pronouncing that right. The prosoma is usually covered by a dorsal shield or a carapace. That's what Shelly's little cover is right here. This is where they'll have the chelicerae. They'll have pedipalps, which are also mouth parts. They'll have 4 pairs of walking legs, and then they'll have medial simple eyes, and then they'll have lateral compound eyes. We'll talk about what compound eyes are a little bit in the horseshoe crabs. And then they have the opistoma, which is the back end region of the animal. In terms of gas exchange, almost all of them either have book gills, book lungs, or tracheae that attach to those for respiration. Let's talk about the sea spiders first. This really weird, sometimes very beautiful group of organisms called sea spiders. These are not spiders. They might be related to arachnids and other arachnids like spiders, but they are not spiders. They're marine. There are about 1,000 species of them. They live in the sediment, on top of the sediment in the ocean, so they're benthic. But they can be in shallow regions or they can be in very deep parts of the ocean. For the most part, these animals are predatory, but some of them are ectoparasites of other animals and very distinct morphology with these really, really slender bodies and really slender legs. And because their bodies are so slender, they have some of their organs that go into their legs, which is really cool. A little bit on their morphology and their anatomy. They don't have tegmata like other chelicerates, which are these fused body regions that we've talked about before. They can have a really prominent proboscis like this illustration here that's with the mouth that's at the end of it. On the cephalon, which is the head region, they have a pair of chelophores, a pair of palps. These are all different appendages that are involved in feeding. They'll have a pair of dorsal eyes, and then that's also where they'll have the first pair of walking legs. Also near the cephalon, they might have these things called oviger appendages. These can be involved in a lot of different behaviors in sea spiders. They can be used for grooming, for food capture, for mating, and we'll talk about how they're involved in brooding of the egg mass in a little bit. And then they have the trunk region, which is the back end here, illustrated here. And on the trunk, they have 4 pairs of walking legs. And each of these walking legs has 9 segments. The anatomical terms that are used to talk about walking legs in chelicerates, and then also in the hexapods are the same kind of terms we use for bones in the human body. So we got the coxa, femur, tibia, tarsus. Well, tarsus we don't use so much in humans, and then they'll have claws that are at the tips of those feet. This GIF here shows it walking, but they use their legs mostly for clingings to stuff rather than locomotion. They don't really move around a lot. And then you have this posterior dorsal tubercle or abdomen at the end here, and that's where the tip of the anus is. These animals, when they feed, they'll cut into their prey with cuticular teeth that they have near the mouth, and then they'll use their sucking proboscis, if they have a proboscis, to extract fluids and tissue from their prey. Sometimes they'll use their chelophores, which are labeled here, to pull off bits of the prey and pull it apart before they suck it in through the proboscis. And in these organisms, they do gas exchange directly through their exoskeleton, so they don't have the book lungs or the book gills that we talked about earlier. And these are just some SEM images, scanning electron microscopy images of some of their crazy mouth parts, showing some of these cuticular teeth around the end of their proboscis in a few different species of sea spiders. Oh, and the class that I didn't even say yet is Pycnogonida. That's the class for sea spiders, Pycnogonida. In terms of reproduction, sea spiders are dioecious, so they have separate sexes, and they also display sexual dimorphism. Like I said before, their bodies are really, really slender. All these are pictures of males, but their bodies are really slender, and so in female, the gonads actually extend out into the legs. And then their gonopores, which are basically their genitalia, are on the coxa of the legs because that body's so slender. Let's see, I said that. Oh, and the gonopores are ventral on the body. And then the females will release the eggs out of those gonopores, and then the males will actually collect them into brood masses that they hold with their oviger. So part of the reason that structure that's in the cephalon region is called an oviger is because it's for holding eggs. And the males actually will take care of these broods, so they'll carry the eggs around with them. Even though the female lays the eggs out of their gonopores, the males then collect them up into masses and take care of those eggs. So the males are the ones doing a lot of the hard work parenting in sea spiders. So these are all males that are pictured here holding these eggs with their ovagers. Now let's talk about an extinct group. In your textbook, it has this group as well as the horseshoe crabs classified as Merostomata in their own class. Recent research has shown that these actually belong within the class Arachnida, which is why I've written it this way. So keep that in mind for the exam, that actually, and that's one of your learning goals, is that the Eurypterida and the Xiphosaura are actually within the class Arachnida. So these are an extinct group called water scorpions. They're really, really cool looking. They've been found as marine fossils. They've been found in estuary environments, which are where the marine meets freshwater environments, and they've also been found in freshwater environments. It's suspected that they might have been amphibious, so they might have been able to come up onto land. Most of them were about 20 centimeters, but some of them were up to 2 meters long, making them the largest arthropods that's ever existed. This group's extinct, if I didn't say that already. Now let's talk about the Xiphosaura. These are the horseshoe crabs. There's our little buddy Shelly again. Horseshoe crabs are marine and they're benthic, and they're amphibious because they'll come up onto the sand to mate, which we'll talk about in a second. Horseshoe crabs can live for nearly 20 years, and they max out at about 60 centimeters in length. They're only found in Southeast Asia, Eastern North America, and Indonesia. And they burrow in shallow mud, and they'll eat small animals that burrow into the sediment. And you could see Shelly trying to bury herself in the sediment in this video. Let's talk a little bit about their morphology and their anatomy. The two distinct characteristics of the order Xiphosaura are that they have book gills. These are appendages on the oposoma that are flattened, and they function in gas exchange. And then they have a telson, which is this terminal spike that they have that comes off the very posterior of the animal. The prosoma, on the prosoma, they have a pair of chelicerae that we talked about before. Here they are labeled here. They have a pair of pedipalps, and then they have 4 pairs of walking legs. They also have a gnathobase, which is the thickened proximal region of the leg, and they use it like crushing a jaw for feeding. So that's here, and that's located dorsally. And then they have, on the oposoma, they have 6 pairs of flattened appendages. They have, the first pair covers the gonopores, and then the other 5 are the book gills that you see here, and they use these for respiration. They have this really large cephalic shield or carapace that covers the prosoma. They have medial simple eyes that are found here, those little spots that you saw on Shelley, and then they have these larger compound lateral eyes. And we'll talk about the structure of those in a second. They also have some simple eyes that are near these compound eyes. And so the simple eyes mostly function in detecting light and dark, and then the compound eyes are used more for vision. Their circulatory system is technically open, but it's a little more organized than most arthropods. We're not going to talk a lot about their circulatory system with this group. In terms of reproduction, they're dioecious. They have sexual dimorphism as well. The males tend to be a little smaller than the females. Their mating behavior, what they do is that the males will attach themselves onto the backs of the females, like you can see in this photo here. And then the females will lay eggs in a sand pit that they've dug, and then the males will externally fertilize those eggs. And the females can lay tens of thousands of eggs, and you can see these mass spawning events. I had originally planned an extra credit trip to Delaware Bay to go see these spawning events because it's at the end of the semester, but maybe some other time we can all go to the beach and see these horseshoe crabs spawning. The developing embryos of these horseshoe crabs will stay buried for about 2 weeks, and then they'll come up out of the sediment to go live in the ocean. And the larval stage of horseshoe crabs is called a trilobite larvae because they look a little bit like trilobites that we talked about in previous lecture. And this is just a developmental, looking at the developmental stages of horseshoe crabs. Horseshoe crabs are really, really important for scientific research and for human health. They've been used as a model system in vision research. And some of the reasons that they've been used are because the animal is really large and hardy. Their retinal neurons are really big, and they're really easy to access and dissect. And their visual system is really compact. And has been studied a lot by scientists, so there's a lot of research into their vision systems, and this limulus, this horseshoe crab, has been really essential for understanding human vision as well because they're so easy to study. And then the structure and function of their eyes is actually modulated by a circadian clock, so it's based on daylight, a lot like our visual system as well. This is just a picture of some of the ommatidia that make up the compound eye in horseshoe crabs. So it's mostly the compound eye that's been used for vision research. And these are just the little tiny ommatidia that make up that compound eye. We'll talk about ommatidia when we talk about hexapods. They've also been really, really important for developing these tests that look for endotoxins in various things with health applications. So in their blood, they have these, this is just a picture of the circular system of a circulatory system of a horseshoe crab. The amoebocytes that are in their blood will produce an instantaneous and visible reaction to any endotoxins that are in a inside of mostly liquids. So the endotoxins from gram-negative microorganisms will initiate a clot formation in the blood when it's encountered by the horseshoe crab, and then it causes that blood to coagulate. And so it immediately detects these gram-negative endotoxins. And so they are used very often in testing pharmaceuticals, intravenous drugs, insulin, vaccines, recombinant drugs, implantable medical devices. Almost all of them go through this thing called a limulus amoebocyte lysate test, LAL test, to see if there's anything toxic in it before it's used for human purposes. And what they do is that they have these limulus all lined up, and then they drain their blood, which happens to be blue, from them. It used to be an incredibly unsustainable industry because these were harvested by the thousands from the wild for these tests. But this new paper just came out yesterday. This new paper just came out yesterday that they've now developed a sustainable harvesting method where these can be reared in farms rather than harvesting from the wild, which is really important for their conservation and for the human health applications. Okay, so that's the sea spiders and the horseshoe crabs and the water scorpions. Now let's talk about some of my favorite groups and look at really, really, really cute pictures of them. So I'm just going to kind of march through each of these groups. I'm not going to go into a lot of details about each of these different orders within the class Arachnida, but I do want you to be able to recall them and recognize them for the exam. So this first one is the order Scorpiones, which contains the scorpions, some of which glow in the dark under UV light. I love scorpions. We've also got something that you may be less familiar with. These are the Europygids, or the Thelyphonidids. This is the whip scorpions, or the vinegaroons, and these are called vinegaroons because at the tip of their tail they can shoot out acetic acid, which is basically vinegar, in defense. They're commonly kept as pets. I've always wanted one. Maybe someday I'll get one. They make great pets. You can also have these tailless whip scorpions, the Amblypygids. Don't have a tail, and though they look very terrifying, they are harmless to humans, and they also make great pets. You can see somebody holding one right here. Maybe someday I'll get one of these too, as a pet. Another more obscure group are the Ricynuli. Not sure I pronounced that right. These are the tropical eyeless arachnids. Very cute little guys. Soil-dwelling arachnids, and as their name, as you can see here, they're tropical and they don't have eyes. Very cute. You can also have, there's also the pseudoscorpions, which are another order within the class Arachnida. These are really, really tiny. About the largest they can get is probably about the width of your pinky nail. These are tiny soil-dwelling arthropods. If we had been able to go out into our lab and do a Berlese sample from some leaf litter, we probably would have found some of these. Some of them are phoretic, which means that they'll hitch a ride on other animals. This is a little tiny pseudoscorpion. It's not a parasite, it's just hitching a ride on this fly. They're very, very cute and very tiny. Called pseudoscorpions because they kind of look like scorpions, but they're in their own order, separate from scorpions. Then we have these wind scorpions. There's a lot of weird memes about these things being super dangerous. They live in desert regions, and a lot of troops come in contact with them while they are stationed in Iraq and Iran. They are not dangerous to humans. They can bite, but it's very, very rare that they will bite. So there's a lot of misinformation about them on the internet. This is just one of those desert ones. And then there's also one called a teddy bear sulfuge. That's all furry and cute. This one, these are native to Namibia. You can also have the opalones. Still not sure I'm pronouncing that right, but these are the daddy longlegs or the harvestmen. These are just some really cute examples of ones that I like. This is a European species, but this morphology is more similar to what you might see in daddy longlegs around here in western PA. There's this really cute one in the tropics called a bunny ear daddy longleg that's very cute, and some of them have really striking patterns. These ones also glow in the dark. And then there's the ticks, the Acari. This is one of my favorite ticks. This is a large velvet mite. And if any of you guys follow the Oatmeal, it's an online comic. He did a whole comic bit about the red velvet mite. Fair warning though, it's kind of gross. It's very cute and gross at the same time, which I think summarizes our invertebrate zoology class really well. This here is a rhinoceros tick, and this is a zebra tick. Ticks live on their parasites. They can live on a lot of different organisms. These are some ticks that are feeding on the blood of a lizard. There's an engorged deer tick for you. And then these are some mites. This is a beneficial mite that you can buy in order for biocontrol purposes, to eat pest species of mites and other pest species that might be living on your plants. And some mites are phoretic, like those pseudoscorpions I talked about. These are a bunch of mites that are hitching a ride on a bumblebee. So I've seen a lot of these mites in my time studying bees. These are some orders that are not mentioned in your textbook, but I thought we might as well just go through all the orders since we're bringing them up. These are the Palpa Grotty, or the micro-whip scorpions. These are some pretty small soil-dwelling arthropods that have that cute little micro-whip at the end. These guys don't have book gills or book lungs. They're really small, and so they just respire straight through their exoskeleton. You also have the short-tailed whip scorpions. These are some other small soil-dwelling arachnids that you can find. The Schizomimida. And then last, but certainly not least, are the Aranai. These are the spiders. There are about 36,000 species of spiders. Again, still not sure I'm pronouncing that right. This is a picture of my pet jumping spider Boop cleaning herself. This little video here. Some of you got to meet her before she passed. This is a picture of a peacock spider doing its signature mating dance. Here's an orb weaver spider. What else we got here? These are some really great pictures taken by Thomas Shahan. I'll show you a link to him, his website here. These are some smiley face spiders from Hawaii. Here's a tarantula. Some spiders are social, and they're a really, really diverse, really interesting group. So let's talk about class Arachnida as a whole and some of their defining characteristics. First you have the prosoma, which is at least partially covered by a carapace within this group. And this is another really great illustration by Thomas Shahan of some of the diversity within the class Arachnida. The oposomal appendages are absent or replaced by spinnerets or pectines, which we'll talk about in a second. And then gas exchange is accomplished via book lungs, tracheae, or both. So let's talk a little bit about their morphology anatomy, and we're going to use this generalized image of a tarantula to talk about arachnid morphology. On the prosoma, they have their chelicerae, their pedipalps, they have their walking legs, which have that same terminology for describing the parts of the walking legs as we talked about before. They have that dorsal carapace that we talked about. And then they can have a multitude array of compound eyes. Thomas Shahan has another great illustration showing all the different eyes combinations that you can have in spiders. The joint between the prosoma and the oposoma is called the pedicel. And then on the oposoma, you can have these ventroterminal spinnerets. This is where they will extrude silk. And some of them have pectines, which are sensory features that are found in scorpions. On scorpions, a stinger is found at the end of a telson. That telson is a modified appendage that you only see in scorpions and other things that have scorpion in the common name. They use their chelicerae in this group to tear apart prey, and then they may also be able to deliver a poison from the tip of their fang on the chelicerae. And then they'll also have pedipalps that aid in handling the prey. There are ventral lung slits that act as spiracles that lead to the book lungs that you can see here. There's their lung slits. And then the book lungs connect to a system of tracheae that will aid in respiration. In terms of reproduction, like all the other ones we've talked about so far, they're dioecious. They can have 1 or 2 gonads in the oposoma. The gonopore is posterior to the lung slits that we saw in that last picture. And most males will deliver a spermatophore to the females for internal fertilization. Parental investment is very common. Here's a scorpion taking good care of its babies. And a lot of arachnids, particularly jumping spiders and other spiders, will have really elaborate mating dances that they do for females. KQED Deep Look has 2 really, really great articles and videos about mating behaviors in spiders. This is a really great one showing a spider that's singing for the female to try to entice her to mate with him. These are some cutie jumping spiders. And then here is another Deep Look article from KQED that talks about how this is a male black widow spider destroying the web of a female spider to stop her from mating with other male black widow spiders. And that's it for this lecture. Even though I spent a lot of time on it, it's a little shorter than some of the other ones. I just really wanted to include a lot of pictures and try to encourage you to think about arachnids and other chelicerates in a new way, because I think they're really cute. A lot of people think they're really cute. Jumping spiders particularly are super cute. And I also wanted to encourage you to check out Thomas Shahan's Twitter page and look at some of his amazing, beautiful artwork. He's one of my favorite artists and photographers of spiders and other chelicerates. And that's it. That's all we've got for exam 3. Next time, we will talk about obviously my very favorite group, the hexapods, because insects are in there. Okay, bye-bye.